Hey guys, so here we are ready to do part 2 of the Diffusion Through A Membrane Lab. Remember, this is a New York State Mandatory Lab, so when you get to part 2, you should make sure that you read through this. I'll be reading through it in class with everybody. I'm not going to read it in the video, and it will be your responsibility to do that kind of thing. Basically, what we're going to start with here is we're going to be looking at red onion cells. So that's what I have here in a little baggie under your microscope. You're going to need, and of course, if you're home and doing this virtually, you're going to watch me. You're going to have a slide, a cover slip, and a little piece of red onion. So what we're going to do, we also will need the microscope and all that kind of stuff. So what we're going to do here is we are going to take a piece of this red onion. So if you remember from the Comparing Plant Animal Cells lab, we took the inside curve. We were looking at the inside, peeled off. You can actually see the little loose piece of epidermis here. We peeled that off to look at that under the microscope for the comparing plant animal cells. This time, we're actually going to use the outside because we want that purple bit. So what I'm going to do is I'm going to use my complete lack of nails here, and I'm going to peel off a piece, if I can. Maybe I'll try this side. Not having a lot of luck over there. It is a little tricky. Usually once you get one piece, it's like good to go, but it takes a little bit to get this piece here. All right, I got a little bit. I got too much of the onion, but I can cut that part off. Okay, so here we go. Can you see how it is getting peeled off into that one little thin? Ooh, that's a lot. It just keeps going. There we go. That's fine. That's a really big piece. If I had pulled this off in class, I would probably give it to 2 or 3 kids. I'd be able to break it in half. I'm actually going to break this part off because I had to pick off a chunk of the onion under my nail to get it. And then I will just take this. I'm going to put it on the slide. Just smush that onto the, get off me, onto the paper towel. Okay, so now I've got this piece of red onion on the slide. We want it to be, again, just like the last time, we want it to be as flat as possible. I'm actually going to open this up in this because I think you'll be able to see it better in a zoomed in version. Maybe. Maybe not. There we go. It's used. Oh, okay, because I'm using it for the recording. All right, so I guess, let's see, can I make it bigger? I broke it. Where did you go? Oh, look, I don't know how I did that. Here we go. Okay, so I wanted to use it in this program because I wanted to be able to zoom in on it a little bit closer. So here's my piece. The glare is a little tough, but here's my piece of red onion on my slide, as flat as possible. It's probably not going to be perfect, but that's all right. I'm going to take a little bit of water, which is what I have here in this beaker. It's just water. It's in a kind of a dirty beaker, but it is just water. It also is a little bit on the warm side, which is why it kind of looks a little cloudy. But I'm going to kind of gently, just like I did with the onion, the other onion, the white onion, gently put some water over the top. And I want to be kind of quick about it. Once I put the water over the top, I got to get my cover slip. I'm going to pop that bubble there and put the cover slip over it. Otherwise, your onion is going to curl up, and we don't want it to curl up. So here's my wet mount of my red onion. And if you look really close, you can see it's got a little bit scratchies in it, but that's okay. Okay. So then we're going to put it onto the microscope. So open up the microscope program. I'm going to start with it on scanning power, as always. I will fix this. Of course, it always takes a few minutes of adjustment when you first put something on the microscope. So I'm going to put it under the microscope. I probably have to adjust the white balance. Ooh, that's a nice big scratcheroonies. Okay, let's see. White balance it. I'm gonna increase the light because, oh, that's a lot of light, but it's a little dark. These big circles here, as per usual, are air bubbles. We don't care about those. I'm gonna try to find a little different spot. Less air bubbles. Oh, there's more air bubbles. Okay, so let's look at, come back. Very hard because the camera is not facing the right direction. And so, all right, that's good enough. Here we go. We're gonna stick with it. It's harder than I thought. So we have a line on our slide. That's what that line is there. So don't worry about that. A really big air bubble that kind of looks like an egg cell, actually. Looks like, but it's air bubbles. And then we have these beautiful red plant cells. So they are similar looking to the onion cells we looked at the last time, but the onion cells we looked at the last time looked like these guys that are empty. There's no color in them. But we want to be looking at the red ones. Okay, so this is what the red ones look like on low, or I'm sorry, on scanning. I'm gonna move it up to low power. You can still use your coarse adjustment on low. You just have to be careful. And it might take a little coarse and a little fine. Let's see, here we go. Try to get those, there we go. Those are pretty cells. Okay, so this is on low power. Now remember, my microscope, I am actually using one of your guys' microscopes this time, but it is still attached to the camera that it connects to the computer. So I still think that there's a little bit of extra magnification. So if it looks a little bigger for my power than you guys in class looks on your low power, it's probably the camera is adding some magnification to it. So here's these beautiful, beautiful cells. Moving it the wrong way. I want to center them a little bit more. There we go. Nope. Okay, I guess we'll center these cells. That works. Okay. So we're going to take a look at these cells. Now I'm going to bump it up to, now that I have this nice cell centered, we're going to bump it up to high power. I definitely need more light. Really give it some good light here. And now remember that you're only using your fine adjustment knob on high power. You do not use the coarse adjustment knob on high power. We're seeing probably like a bunch of junk on the slide and the cover slip, but we are also seeing the animal or the plant cell, the onion cell here. So you're only seeing half of it because you're so zoomed in. And you can see that it's all pink. Here's another one here that doesn't happen to be pink. So that's what you're seeing. So in your lab, the first thing you have to do is copy and paste a picture of what you see from your microscope banks. This is just like the other one where we did, where we had a bank of pictures. You can look at the bank of pictures that I have provided you here. So you can take a peek through. And this picture here is looking for you to draw and check, draw. It's really draw, obviously. In the COVID world, it's copy and paste. But we're looking for low power, okay? So I'm going to put low power in here so that you know that it's low. Low power. Okay, and then your job, again, is to look at your picture bank and determine which of these looks the most like our slides did on low power. Okay, so this, let me pull that back up again. This is what we have on low power. I gotta lower it. There's way too much light now. There we go. Bring it back into focus. This is what we have on low power here. So you want to pick the picture out of your picture bank that looks the most like that. Okay. Paste it right here. Then we're going to move on. So now we're doing a similar thing that we did with the stain when we stained our onion cells with iodine. I'm going to take the onion cell off the microscope, put it back underneath here. I need a piece of paper towel like I did the last time. I probably will need a bigger piece. I'm going to put it along the left side of my slide and my cover slip. Immediately, you can already see that it's starting to soak up some of the water. And then I'm going to use another beaker that I have here that is filled with a salt solution. So it is not just plain water, it is salty water. I am going to add a salt solution to, let's scooch over. Add a salt solution to this side. Again, just like the last time, we want to make sure that we kind of do it gently. We don't want to do too much at once, otherwise we'll flood out the slide. Give it a second. And then you can see that it's sucked up some of it. Sometimes I switch the side of the paper towel because the paper towels are kind of junky. Can do another roll of it here. Try to make sure you get your cells good and salty. There's a really big drop of water on top. Okay, so once you've done that, you're gonna take it off, not take it off. You're gonna put it back on the microscope. And again, you're always gonna start at scanning power. Scanning power. Let me show you my microscope program. It's way too much light. The lower you are, the more light you get. So you have to kind of play around with the light. And here we are. Those are actually perfect if I can just get them centered. What makes it do that? Okay, so that way. And this way. Nope, too far. Okay, so these are perfect cells that we want to look at. This is the kind of thing you're going to see. So these ones pretty much look the same way they did before, but these ones clearly look different. You can see here is the outside of the cell. That's your cell wall. And you can see that the cytoplasm and the cell membrane have slunked away and gotten pulled away from the edges of the cell wall, and the cell membrane has really shrunk. The reason we use the red onion is because you can see it so clearly in that one. Let's see if we can find another good spot to look at and zoom in on. Okay, so here's another great couple of cells here. And again, sometimes some of the cells take up more of the salt than others. So like these guys here look pretty much the same way they looked before. They look like they probably didn't get too salty. But this guy here, actually I think this might be the exact same size. I was just looking at this guy. This guy here, they clearly have pulled away from the cell membrane, has pulled away from the cell wall, and it really has shrunk up, which means that the water in that cell must have diffused out, causing the cell membrane to shrink. Okay, so when you look at your lab, you're going to observe, you're going to answer that question, and then we are going to look at your slide, and again, we're going to look at it, this time we're going to look at it on high power. Okay, this time we're going to look at it on high power, I believe. Okay, I'm going to put high power here. So let's switch here. We're on scanning still. Let's pop it up to low. We probably have to add a little bit of light. Take a look at that guy. This guy here really is very nice. I'm going to add just a little bit of, too much light. A little bit of light. Might balance it a little bit. This is an air bubble. This down here is still an air bubble. Air bubbles are the story of my life. I just want to center it a little bit more. Oh, God. All right, we'll use this guy. We'll just use this cell. That's fine. Okay, that's another good example. So we'll use this cell. The other one, I lost it. So you can see, again, the cell membrane has really pulled away from the outer edge of the cell wall here. If we bump it up too high, I'm definitely gonna have to add some light to it. Much greater exposure on high. And again, remember that on high, you're only using your fine adjustment knob if you were really using the microscope. So he's a little off center. I don't want to move him because I'm afraid I'll lose him entirely. But you can see here's the cell wall. And there's all this space now, and the cell membrane has really pulled away from the cell wall. Again, this is because the water is diffusing out of the cells because the cells have been put into a salty environment. So you're going to look for the picture in your microscopic cells bank that shows this on high power, and then you can double click into it and label your parts as well. Okay. The last part here, you have to answer this question. Then the last part is basically going back and replacing the solution on the slide. So taking your cell or taking your onion off the microscope, putting it back on here, getting another piece of paper towel. And this one is a little bit harder. I'm going to go back to the water, not the salt water, because it's a little harder to flush the salt water out. But basically, we're going to try to flush the salt water out. See if we can get the cells to go back the way that they were. And this one, you're really going to be a little aggressive with. So I'm going to let that sit for a second, see if we can soak some of that up. Pop that bubble. Soak it all the way through. You can see the water will move. You can actually see it happening. And hopefully we can get our cells to re-expand. This one is, like I said, a little tricky. You really got to flood it to really flush all the salt out. And so sometimes when we put it back on the microscope, we're going to find that it actually is kind of a mixture of both, although it was sort of a mixture of both the last time too. But it's going to kind of continue to be a mixture of both types of cells. You're going to see some cells that are clearly salted because the cell membrane is clearly pulled way far away. Oh my goodness, I'm making such a mess here. Way far away from the cell wall. That's what I mean by flooding the slide. That's right there is what I'm talking about. And then you're going to see other cells that look kind of like back to normal or are normal or whatever. So we'll go back to our scanning power. Take a look back at the microscope. We'll lower the exposure. And let's see if hopefully I didn't flush out all of the pink, which sometimes happens. Let's see. I don't know what that is, but these cells look pretty good. We're on scanning power now. Yep, so you can see they've re-expanded. They're still a little almost unsure about it. I don't know what that is. It's just junk on the slide. Yeah, that's just junk on the slide. So yeah, so these guys here, you can see that they have sort of refilled. Looks like they're a little upset about it still, but they are, like, you can see some of these parts are still kind of dark pink. It's still kind of readjusting back to having normal water. If we look at it on low power, it should look pretty similar to what we were seeing before. Like I said, you can still see some spots. Here's a cell that still has some salt in it. So it's not quite reconfiguring its way back to normal, but here's a nice normal cell with the cell membrane has gone and expanded back to where it belonged. And then on high power, increase the exposure. And again, only the fine adjustment knob on high power. And let's see what we got. We are looking at just the corner of one of those cells. Actually, it's the cell that hasn't fully expanded back yet. So there's still some space here that the cell membrane hasn't fully stretched back yet, but it will. I'm afraid to move this because I know I'm going to mess it up, but okay, so here's it's only the top part of the cell because you're not going to be able to see the whole cell. But here's the top part of the cell with a whole cell membrane that has re-expanded to fill the space. Here you can see that it's pink all the way around. So that's what it would look like on high power after you re-put the water or the distilled water back on it. So again, this is that last picture here. This is also going to be on high power. And you're going to use your microscopic cells bank to pick which picture looks like what you want it to look like. We'll be mostly working on the questions together. I'll let you do some of them on your own, and then we will finish them up together. If you have any questions, please feel free to let me know. Good luck on your state lab.